Good morning. Today's series is on chapter 23 on natural selection, and it focuses on how we get the genetic variation that ultimately ends up allowing a population to evolve. So there are three characteristics that we look at when we observe a species that has evolved in two different directions. So, for example, I'll give you animals mice and fish that have been studied extensively that are the same species but have evolved to have different genetic traits, and the reasons behind that, because ultimately that is what allows evolution to happen. So the number one quality that is needed for evolution is to have a good population, or at least an average population of heterozygous individuals, because those will have the ability to have a completely dominant characteristic or a mix or the recessive. And the variation between populations, for example, there are numerous studies that have been done on rodents. And one study was done on an island where they had a pretty heavy mice population. And they had a volcanic, truly a volcano, start to develop in the middle of the island. I'll stop and tell you a story in South America where there was a farmer, a corn farmer, very poor, in a remote village, who had started noticing that part of his cornfield started to rise up. And he went to the village and got the priest, because he was worried that perhaps it was a religious thing that was going on. And the priest, who was reasonably educated, went out to his farm and then went back and tried to contact some geologist and volcanologist, because he suspected that what was happening within just a very few short days was that a volcano was starting to develop on this poor man's property. And in addition to that, all the corn was dying. So what was happening was, sure enough, the magma was rising up and underneath his cornfield, and the gases from that were killing his corn. Well, something happened very similar to on another Philippine island, where this volcano started to emerge. The magma emerged right in the middle of the island. And what happened was that it divided a species of mice. And so one ended up on one side of the island and the other ended up on the other side of the island, trapped from each other because of the volcanic activity right in the center of the island. And scientists did DNA studies on the mice and found out that they definitely were from the same common ancestor, but one developed a totally different coloration eventually than the other group of mice, and that had to do with their habitat. The same thing sort of happened in another area where, again, volcanic activity, and where there was an eruption, and the black obsidian, or the volcanic glass, ended up hardening in one area, and right beside it was desert. So we have this obsidian, black obsidian, and then a foot later, white desert sand. And again, with a rodent population they studied, and they saw that due to natural selection, and just like the peppered moth, over decades, those mice that chose to live in the volcanic area, the obsidian, those who had a darker coloration naturally were the ones that survived from predators, and the ones that chose that were living in that desert habitat, that lighter coloration, had the better chance of survival. So that just a few decades later, we have a original species of mice, one that is black and one that is white. So again, that is from that variation from population due to an environmental change. Another variation that happens is called a cline, and the textbook on chapter 23 discusses a type of fish. I always call it the michicanga, because I asked Mr. Nimizek once, who a former teacher at Pelham High School, how to pronounce it. He was a marine biologist and worked at the New England Aquarium and for the state of Massachusetts in their fisheries department. And he said he couldn't pronounce it, so I just call it the michicanga fish. But it's a species that we know from DNA evidence. It's discovered along the entire eastern seaboard from Maine to Florida. And when they do studies on this fish, the michichangas of Florida, if you capture them and place them over time in water that changes temperatures, if you were going up the seaboard to off the coast of Virginia and off to Maine, they perish. They can't survive the temperature variation. And the same happens if you took the main species and over weeks gave them a temperature decline. They also die. And they discovered that there was a genetic variation in them, an alteration in a gene that causes an enzyme change in them that allows them to adapt to different water temperatures. So the Florida Michichunga has their enzymes only function in a very warm temperature, and the Maine Michichungas only function in a very cold water temperature. So same species of fish with a mutation that allows them to live in two very vastly different environments. So again, genetic variation, much better chance for variation in a population that has an average amount of heterozygotes. And that variation in populations can be due to environmental changes, everything from temperature or the landscape, etc. So we have four major sources of genetic variation. One is formation of new alleles. And of course, that would come through a mutation. So in previous chapters, we've talked about mutations in frame shift mutations in the DNA, or a missense or a nonsense, and all of those causing rather dramatic mutations, often causing death of the cell. But there are those mutations that end up with organisms that survive, and sometimes those new allele changes are beneficial. Obviously, in the case of the peppered moth, where previous to the Industrial Revolution, the whiter moth with the black specks had a much better chance of survival over the one that had a slight color mutation that was grayer. And then that reversed during the Industrial Revolution. Remember that the color change is called industrial melanism. And when that changed, we ended up getting a species that had benefit to be the grayer color. After the Clean Air Act in England, that was fairly early on, they then saw the reversal back to the white peppered moth. So another factor other than formation, number one, formation of new alleles, is second, an altering of a gene number. Again, very similar to the formation of a new allele. We could have a deletion or a disruption or a rearrangement. And again, at times that altering of the gene number can be beneficial to a species. Third, we can have very rapid reproduction, and so we don't see that so often we don't see that at all in humans, but in other populations we do, especially insects or bacteria or viruses, that we can see a very rapid reproduction of a new genetic variation. And focusing on viruses, because that's probably the easiest to understand right now, is that viruses that are not considered to be alive, even though they do quite a bit of damage, they don't meet the characteristics to be alive. They do not respond to stimulus. They can't reproduce on their own. They must have a host. They don't have their own energy source, and they don't create a waste. And yet, as they are using a different host to be able to reproduce, they often get very quick mutations. And that's what can be very difficult to treat. HIV did that, where it's early on, it kept mutating and mutating and mutating. And so as physicians found treatments for people that were suffering from HIV, no sooner would they come up with a mutation than the virus mutated. No sooner did they come up with a treatment than the virus mutated again. In addition to that, if we look at bacteria, early on, we had a treatment for a Staphylococcus aureus, and that was penicillin. And 2 years after, and staph can be quite dangerous. It's found on our skin, it's found everywhere, but if you're ill, that can be a real problem for you. And so they discovered that the Staphylococcus aureus, just after 2 years of using with penicillin, became resistant to that. So that was a mutation. And that, with rapid reproduction, causes more mutations. And fourth, the source of genetic variation is sexual reproduction, where we're getting those different alleles from different parents. So remember, 4 different sources for that genetic variation: formation of a new allele, an altering of gene number, that deletion or disruption, rapid reproduction, and sexual reproduction, where we're getting 2 different alleles, one from each parent. And the opposite of studying evolution were 2 mathematicians, Hardy and Weinberg, who studied What happens if a population doesn't evolve? Can we prove if a population is not evolving? And again, they were mathematicians, so they came up with a criteria of what would be needed for a population not to evolve, and then, of course, a mathematical formula to prove whether it was happening or not. So there are 5 requirements to the Hardy-Weinberg formula. Number 1, for a population to not evolve, there can't be any mutations. We must have random mating. If you are artificially selecting a population, then you can control the conditions of evolution. There can't be any natural selection, so any mutation, anything that we consider a variation in a population that would help with natural selection can't happen. You must have an extremely large population, because if you had a small gene pool, there would naturally be mutations. And you can't have any gene flow. And gene flow is discussed as having any population leave, because that would alter your standard population. And we can't have any population entering. So again, because they could have a different gene pool. So we can't have any emigration or immigration. And again, the 5 requirements for Hardy-Weinberg proving that there isn't any evolution in a population is: there can't be any mutations, there has to be random mating, there is no natural selection, you have to have an extremely large population, and you can't have any gene flow, population coming into the population, immigrating, or anyone leaving, emigrating. And Hardy-Weinberg, again, being mathematicians, came up with a formula where you could demonstrate whether there was or wasn't a population change. And so the mathematical formula is p squared plus 2pq plus q squared equals 1. And what does that mean? Well, P is standing for a person or a population that is completely dominant. And so if you had a certain number of cases that you knew were 100% population was dominant, they would be the p squared. If you knew a population was completely recessive, that would be the q squared, leaving pq as being heterozygous. So 2p squared plus 2pq plus q squared equals 1. And let me hold that formula up for you. Let's see if you can see that. So again, p squared equals the dominant population, q squared is the recessive population, 2pq would be the heterozygous. And you could actually be given, you'd have to be given if you were had a set of formulas to work out. If you were given the completely recessive population numbers, or if you were given the completely dominant, then you could figure out what percentage of the population was heterozygous. Again, if you were given the population that was heterozygous, then you could plug in the numbers and come up with what would be the recessive and what would be the dominant characteristics. And Hardy and Weinberg, again, this is a formula to prove that a population is not evolving. And I will put in on Google Classroom a few of the Hardy-Weinberg examples and a video on just practicing how to solve for Hardy-Weinberg. And that is your big characteristics of Chapter 23, or how a population evolves. Again, next video will be on how to solve a Hardy-Weinberg problem. Thank you.